chapter 19, but this is the third PowerPoint lecture that I've put together. I probably haven't posted it for you guys, and part of the reason for that is I want you to pay attention today and take notes. I'll post it after class, though. Huh? On the PowerPoint. Yeah, you could. But then you would have the answers too, because I didn't erase them. So, I don't want you to have the answers ahead of time. I want you to have read in your book. Let's see how many of you guys have actually done that or can pay attention. Okay. So, the next objective, right, or question from our book that we want to be able to try and answer is to describe the immunological role of, does anyone remember what PRR stand for? Yeah. I hate that we use all these acronyms. I meant to write this out. Pattern. Is that how you spell pattern? TT. Yeah, oh, see, it's telling me it's wrong. Pattern. Rec. That's probably the most important part of the word, is that these are receptors, right? So, that recognize specific patterns. So in this case, the two that we're looking at, mannose binding lectin, it tells you what it's recognizing. It's recognizing mannose, which is a sugar. Do you think it's a sugar that's in our membranes? No, because we're not going to recognize ourselves, right? We want to recognize things that are not self, right, that are foreign. So these sugars are found in microbial membranes, things like bacteria and yeast. Does it tell us what bacteria it is or what yeast it is? No, but in general, it tells us a bacteria or yeast, so it's probably a bad guy, right? Because it's not us. The other is C-reactive protein. So this diagram shows mannose binding lectin. The other receptors that we talked about before, like the toll-like receptors, remember they're embedded in the membranes of the cell's membrane or in the endosomes. These receptors are actually secreted. So these are just floating out there. So see mannose binding lectin, right? This receptor, it's just out there floating. But as I said, as the name implies, what it's binding to here is the mannose molecules on, the, say, this bacterial membrane. You see how the little cup is binding? That's a good point, right? So right away, Lance's brain is on this morning, right? So he's saying, well, if mannose binding lectin binds to it, right, then it can't bind to us. And that is probably true if it's the mannose that's binding to us. I don't know if that's the case. But the important thing here is we're recognizing something as being foreign, right? That's the number one thing for our system, is we've got to recognize. And then, of course, we want to respond in some way, right? So what do you see in this diagram? You see two responses, right? So here, right, the response is creating what in the cell? It's creating a pore in the cell. And so if you create pores in the cells, what's ultimately going to happen to that cell? It's going to bust, it's going to lyse. So if you read here, it says activation of complement, which is a series of proteins that we're going to talk about that can do just this. They can cause pores to form in the membrane of microbes that we recognize and cause them to lyse. I mean, we can directly destroy them, right? We have proteins that will directly make pores in their membranes and cause them to lyse. That's pretty cool. The other is something we're going to also talk about as one of the outcomes of the complement system is what we call opsonization. They don't have the term in this particular diagram. But in simplest terms, I always think of opsonization as tagging for destruction. Because what is happening here? This is a bacterial cell, right? Here's the mannose binding lectin attached to it. So that's our system kind of recognizing this is a bad guy, right? It's tagging it for destruction, saying, this one, get rid of. It's bad. Right? It's like when they go through on the side of the highway and they put the X's on the trees they want cut down. Right? We're basically putting a big old X on the bacteria. By the mannose binding lectin binding to it, we're saying, you need to go. How we're going to actually destroy it, one of the ways we can do that is that we have cells that have receptors for these substances. So in the case of mannose binding lectin, when it binds, we have receptors on our cells, like a macrophage, that can grab onto that. Do you see how it has a receptor right there? Receptor for mannose binding lectin. So it's binding to it, grabbing onto it, and again, the first part of it is being able to attach to it. Then it's going to bring it in and engulf it and destroy it. And that's what you're going to read about in your homework, is how macrophages, phagocytic cells, actually do this process of phagocytosis. If you haven't read that far in the chapter yet already. Right? Our last section are the actual cells, right?
So the PPRs are kind of like the cops that attach and bring people
They're... Yeah, I know actually it would be the Crime Stoppers, the people actually calling the tip. You know, that's what these receptors are, right? They're letting the cops know, hey, this bad guy is here, right? Because the receptors themselves don't actually destroy, right? Where a cop could, right? So this is that very first important step of recognizing that it is something foreign, something bad, something we need to find a way to eliminate. And again, notice we're not just sticking with one way, right? When mannose binding lectin binds, the only way we're going to destroy it is complement activation and cell lysis? No. Another way is a phagocytic cell could come along and bind to it and engulf it and destroy it that way. So we're not limiting ourselves even just to one way to destroy something once we detect it. So you'll see these repetitive patterns, right, where we use things over and over again. So these are examples of opsonizing, right? There's that term I used. Secreted pattern recognition receptors. So we're actually secreting them, right? And they're binding. So in the case of mannose binding lectin, it coats mannose-rich surface of yeast and bacterial cells. So as we saw in that picture, right, it actually binds to the surface of the bacteria to the mannose. C-reactive protein, on the other hand, binds to something else. It binds to phospholipids found, and again, phospholipids that are unique to bacteria and fungal cells. So different from our phospholipids. Right? Opsonization, they opsonize, they tag for destruction by phagocytic cells, right, or a complement system. So the next thing we're going to look at is complement, right? So how did activating that system cause the lysis of that cell? So first of all, the first question we need to ask is, what the heck is complement, right? What is it, right? How does it protect us against microbes? So as you watch this animation, I'm going to help hone you in on that question, is I really want you to know there's three main results of activation. There are three things that happen. One of them we've already seen. Right? What is that? When we activated complement, what happened to that cell? It says right here, activation of complement. Yeah, put pores in the cells and lyse the cells. Right? So that's one. Right? So watch for that one. That one's definitely there. But there's two more things, right, that are going to happen when activation of complement happens. There are three ways we can turn on complement cascade. There's the classical pathway. There's the alternative, and there's the lectin pathway, as we just saw on the previous slide again, right? Repeats. Right? So one of the ways we could get this whole thing started is mannose binding lectin binding to it. That one's not discussed in the animation. The animation discusses the classical and alternative pathway. So as it talks about these two pathways, you want to know what starts the whole thing, right? So in the case of mannose binding lectin, what started it all? The mannose binding lectin binding to the mannose, right? So what's the first thing that binds to the surface of the cells that activates classical pathway? And what activates alternative pathway? Okay, so I'm going to play this one, and it does have closed captioning that is correct. So, and to play these, once you download the PowerPoint, you have to be in slide presentation mode. So closed captioning is right down here. And
complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3a and C3b are constantly being formed from C3, but without activation, they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properidin to form a complex called C3 convertase, which cleaves C3 into C3a and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3a is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3a and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properidin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components, including C6, C7, C8, and C9, to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
that was a lot of stuff, huh? Yeah, we're going to go back and step through it. Okay, So at the very beginning, and kind of at the end, it told you those three outcomes, right? Here's one of them that we've already seen with mannose binding lectin, is the membrane attack complex, right, which will actually lyse the cells. The other is inflammation. And then one more. Yeah. Opsonization, right? Opsonization. So those are the three main outcomes, right? So we get an inflammatory response, we get opsonization, and we can get cell lysis by the MAC complex, right? Which stands for membrane attack complex. That's important, right? We're attacking membranes. Okay, let's see who is really paying attention. So that's what happens, right? But how do we get the whole thing started? What has to bind to the cells first for the classical pathway? So let's rewind. Antibodies, right? Antibodies. Antibodies are the first thing that binds. That's what helps our system recognize that it's foreign. We talked about antibodies yet? Not really, huh? Anyone know why? They're a product of our adaptive immune response, right? So remember how we said we talk about these things separately, but they really work together a lot? Here's one really good example, right? The complement cascade is really part of our innate, right? It really isn't, for the most part, cell-specific. But it somewhat can be in the case of the fact that we're using an antibody that is specific. I mean, we could go right down to the strain of E. coli 0157 and recognize it with a specific antibody. That antibody could bind, right? And then it activates the cascade. The good news is you guys aren't in a graduate level immunology class. By no means am I going to make you memorize every step of this process, right? Just the major steps, right? Obviously the major outcomes, the beginning and a little bit in the middle, right? So let's talk about that. So first the beginning, right? Classical pathway, antibody binds, right? And then the proteins in this system, you'll notice they're all labeled C for complement. And then they're numbered, right? And how did they say they were numbered? In the order they were discovered. So does that mean the order in which they're activated in this cascade? And cascade, it's like a domino effect, right? Once one thing happens, the next thing happens, and then so on and so forth. Right? That's why they call it a cascade, right? It just keeps going. It's like dominoes, right? Once one hits the other, they just keep going. So depending on which pathway, right, the numbers don't quite go in order. They kind of do a little bit. Truly is only a couple out of order. It's not too bad. So number one, classical, antibodies bind, right? And then they bind to the complement proteins, right? And we get this sequential turn of events that happen. For the alternative, and so what was the major event, do you think, here for classical? So here's C1 binding, and then C2 being split. And notice when they split the molecules, right, that's when they get the little A and the little B designation. That's to let you know it's a fraction, right, a piece of the original complement molecule. So they're in our blood, right? They spill into our tissue during inflammation because they're in the liquid phase of our blood, these complement proteins. But for the most part, they're inactive, right? You've got to start this to activate them, right? And it's kind of like a tag team match, right? They're like, okay, you're it, you're it, right? So this one cleaves the next one, right? And then the next one. And then we convertase. So what is a convertase? It ends in ASE, doesn't it? So that tells you this is an enzyme, right? And what does this enzyme do? No, watch. This is one of the important ones. Here comes C3. The reason why it's called C3 convertase is it cleaves C3 into C3A and C3B. Right? So again, it's taking C3 and it's breaking it into two pieces, A and B. And they're different, hence the different letters, A and B. So then we jump to the alternative pathway. What binds to the foreign substance first to activate this? C3b. You've heard C3b a lot now, huh? It's an important one. So for classical pathway, it's antibodies. Is the prime substance of our body that binds to the foreign substance that will activate complement. For the alternative pathway, it is one of the pieces of the complement proteins. It's C3b. Which, as they said, we're naturally always breaking down C3, right? But it has to get further activated, right? It has to bind to something, right? Other molecules come in, as they say, right? So factor B and D and piperidin, right? And we convert, again, to an enzyme that's just going to keep breaking down C3 into C3A and C3B. So at this point, all the pathways are similar, right? They all somehow generate C3 convertase. That's going to keep breaking down C3 into C3A and C3B. So one of the important things about C3A is that, and they show it really good in this animation. You see that mast cell right there? He's degranulating. He's releasing histamine. Remember, what effect does that have on the blood vessels in the area? They dilate, right? They're going to dilate. We're going to increase blood flow to that area. Why do we want that? Why do we want inflammation? Why do we want increased blood flow and fluid leaking out? What else is going to come out of that blood vessel? The white blood cells, right? The white blood cells. Because we need them for this next thing, right? That as C3b is being produced, what does it do? It goes somewhere, doesn't it? It's an opsonin. It binds to the surface of the cell. So as we're producing this, it's binding, it's tagging them for destruction, right? By what? By phagocytosis. And who does that? The white blood cells, right? So the white blood cells have got to get there to do this job, right? So this isn't going to happen, really fast. Maybe the ones that are already in the area, like the macrophages, but those neutrophils that have got to travel through the blood, right, come out and then actually engulf that cell, right? We've got to get them there. And then once they're there, we got to make sure that they know which ones they're going to eat, right? So I always remember C3b. I remember B for binding. Right? And there's 3 pathways, right? So we know number 3 is a really important number. And C, as we said, is for complement. But we're not done yet, right? It keeps going. We're not at the end of our domino chain here. We're going to now convert and make C5 convertase. All the pathways have this step. And as the name implies, it's going to cleave C5, right, into the 2 pieces. And these 2 pieces are important too, because C5a is also important for inflammation. It's also going to cause degranulation of mast cells and neutrophils. Basophils. Why did I say neutrophils? Basophils. C5a, stuck on 3. This one interacts with several other of the proteins, of which I'm not going to make you memorize all of them. Instead, what do they form? What is this called? The MAC complex, right? MAC, M-A-C. Not to be confused with a Big Mac, right? So this is some big complex for sure. Again, know what it stands for. Membrane attack complex. So these proteins insert into the membranes, they form this donut-shaped hole, right? These pores in that membrane. So what is complement? It's a group of 30 plus serum proteins, right? So these are found in the liquid phase of your blood that have antimicrobial activities. You notice that there were 9 that are labeled complement, but there were several other proteins, right, like factor B and factor D and piperidin and others. And they get activated by these patterns that we know that are associated with pathogens. They result in inflammation, opsonization, right, that tagging for destruction by phagocytes, enhancing that process of phagocytosis. Or they can directly kill, right, by forming that MAC complex, the membrane attack complex in the membrane. So 3 separate methods of activation. The classical is actually called the classical because it was the first discovered. It's probably not the first one that happens, especially if it's the first time you've seen the pathogen. Are you going to have antibodies against it? No. If it's your very first time you've ever seen this pathogen, you're not going to have antibodies. But this pathway they have found out can be activated by other things found on microbial substances. So that's part of your homework. I'm going to ask you guys on Friday, what are the other substances that can activate the classical pathway? So go back and read. The alternative pathway, we've already answered this one. This is one of those important complement proteins we want to remember, or fragments of a complement protein at that. What activates the alternative pathway? What binds? C3b. And then we didn't watch in the animation, right? But it's similar to the classical pathway. The difference here, it's mannose binding lectin that binds first and can activate complement protein 1. All 3 systems, right, generate C3 convertase and C5 convertase, which break C3s into C3A and C3B and C5 into C5A and C5B. We've already mentioned, right, that C3A and C5A elicit what type of response? Inflammation, right? Because they cause degranulation of mast cells and basophils. What one of the products opsonizes? If I say this 8,000 times, maybe you guys will remember. Or make you say it 8,000 times. What's the opsonizer? Okay, I get about half the class at this point. We'll get all you guys by the end. Okay, and then these guys, right, the C5b, the C6, the C7, 8, and 9, form the MAC complex, which stands for membrane attack complex. And what does this do to the cells? It lyses them. So again, notice up here too, antibodies started it for classical, mannose binding lectin binding to mannose started it for the lectin pathway, and the alternative pathway was the binding of C3b. In this case, they're showing that C3b can bind to things like lipopolysaccharide or lipotectonic acid. Who has lipopolysaccharide, LPS? Gram-positive or gram-negative bacteria? Gram-negative. Who has lipotectonic acid? Gram-positive. So, as we said, inflammation, C3a and C5a because they cause degranulation of mast cells. I've got C4a in parentheses there. Did we talk about that one yet? No, because that one is in the lectin binding pathway. Right? Has the same effect as C3a and C5a. It degranulates mast cells and basophils. You know, causes vasodilation, inflammation. C5b is a neutrophil chemoattractant. So what does that mean? What does chemoattractant mean? It's a chemical, right? And it attracts. And specifically, it's calling neutrophils. And the reason for that is those are phagocytic cells, right? So they're specifically saying, hey, come here, right here. This is where the bad guy is, right? This is where you need to be right now. So all of these are important for inflammation. And yeah, these are some of the few that I expect you to know what they do, other than C3b. So here's a diagram showing basically alternative pathway, right? Because what's happening first? What is this? C3b is binding to the bacterial cell. Right? That is going to help, right, with the MAC complex, right? Because we're going to eventually go down the rest of the cascade and maybe form that. The other thing is going to help us with phagocytosis because our phagocytic cells have a receptor for C3b, right? They can bind to and recognize that this guy needs to be destroyed. So what's the very important complement protein fragment that's an opsonin, opsonizes, targets for destruction? Everybody this time.
C3.
Yes. C3b. So this is a diagram, right, of the classical pathway, right, all the steps. And it's wonderful glory. Right? What's the important things for us? Right? Antibody binds to start it. Right? We generate C3 convertase, which splits the C3 into C3a and C3b. We get C5 convertase, which again just keeps splitting C5 into C5a and C5b. Right? And then we can get the formation of the MAC complex, the membrane attack complex. What they're not showing you here right, is opsonization or the fact that C3a and C5a, right, are going to head out and degranulate those mast cells and basophils nearby, right, and help elicit inflammation. So do we need to know all these steps? No. We need to know it starts with antibody, right? We get C3 convertase, which breaks down the C's. 3's into C3a and C3b. We need to know what C3b does, right? What does it do? It's an opsonizer, right? As well as involved in the rest of the cascade. What does C3a do? That's right. Helps with inflammation because it does what? Causes mast cells and basophils to degranulate, release their histamine. You get C5 convertase. It splits C5 into C5A and C5B. What does C5A do? Same thing as C3A, right? Helps with inflammation, causes degranulation of mast cells and basophils. What does C5B do? It's also involved in inflammation, but specifically it says who? Come here. Come here, come here. Is a chemoattractant for who? Neutrophils, right? We're saying, come here, come here. We need your help, right? Because C3b is binding all the time too, right? Encoding them for destruction by those phagocytic cells. We're saying, hey, get here. Right? And then as it continues down the cascade, the last thing that could happen is the formation of that MAC complex. Membrane attack complex. So there it is right there. Nice, big, beautiful complex of proteins, right? Forms a donut shape with a pore in the center. Here's an electron micrograph. Look at all those dark spots, right? Those are all pores, right? Membrane attack complexes then inserted into that pore cell. He's toast, huh? So lysis of the foreign cells, right? That donut shape, we refer to it as the MAC complex, right? Membrane attack complex. It's made up of several complement proteins. You do not have to memorize which ones, although it wouldn't be hard. Creates the pores. Now this is an interesting statement. Most effective on gram-negative cells, little effect on gram-positive cells. What do we know about gram-negative versus gram-positive? You got peptidoglycan, what? A thick layer peptidoglycan for who? The gram-positive, right? So gram-positive, we know, have a thick peptidoglycan layer, right, making up their cell wall. And where's their membrane? Underneath that layer, right? If you're going from the outside in, you're going to hit the peptidoglycan first, and then the cell membrane, right? Oops. This is a membrane attack complex, is it not? You have to insert those proteins into what? A membrane. How are they going to get to the membrane of a gram-positive cell? They've got to go through what first? They got to go through that peptidoglycan layer first. That's really hard, right? That's not easy. Where gram-negatives, why is it that we can kill them with the membrane attack complex? Their peptidoglycan is not that thick. What else do they have, though? Where's the LPS found? In that outer membrane. Remember that outer membrane that they have? That's what the LPS, right, that very outer leaflet, that outer layer of the outer membrane is made out of. But that's still a membrane, right? These proteins can insert into that membrane. Much easier, right, that outer membrane than having to go through the peptidoglycan thick layer, right, of the gram-positive. So they don't even insert into the plasma membrane, right, because they'd have to get through the outer membrane, through the peptidoglycan layer, which is thinner for negatives, right, to get to the plasma membrane. But in fact, what they insert into is the outer membrane. And that's why complement lysis by the MAC complex is more effective against the gram-negatives than the gram-positives. Right? They've got that outer membrane. So that in this case, that's a good thing for us, right? That we can disrupt that membrane. Does that make sense to you guys? Remember how I promised you gram-negative and gram-positive would never, ever go away, that I would continue to talk about it all semester long? Wasn't kidding, was I? Okay, so the last one for molecules of our innate system are the interferons, specifically the type 1 interferons, commonly abbreviated IFN. So again, this is molecular detection, right? Where molecules are going to help detect and help us decide what we need to do. So I have one more stolen animation. Right? For us to see what happens.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
That's pretty cool, right? So basically, they get infected and they scream to all their friends nearby, look out, right? The bad guys are coming. Get prepared, right? So there's like, what does your book say? Like 300 different genes that get activated with interferon. Okay. So this is their chemical message, right? Interferon is their chemical message. Viruses are here. Get prepared. What do you think happens to this cell that the virus got into? It dies. Because what did it just do? What was this last thing right here? It broke down messenger RNA. Right? When the virus gets in, it needs to replicate its proteins, right? Maybe even its DNA or its RNA, depending. We shut down cellular processes by literally destroying messenger RNA. So if we shut down cellular processes, what's going to happen to that cell? It's going to die. Yeah, it's going to die. But what did not happen? So a virus goes into a cell, it does what? It replicates itself. Did it replicate itself? No, we stopped the cycle, didn't we? But we sacrificed some cells along the way. Right? But as a multicellular host, can we spare a few cells? A few thousand? Yeah, we usually can. Right? A few cells dying is better than them all dying, right, from a viral infection. So your book goes into a little bit more specifics. Right? Interferons A and B, alpha and beta, their primary action is to even inhibit ribosomes from binding. So again, we're going to shut down protein synthesis in the cell that's been infected by a virus. We're not even going to let the ribosomes bind to the messenger RNA. Right? So the host cell doesn't produce any proteins. Neither can the virus. And this is going to cause death of the cell. And it's a different form of death. We refer to this as apoptosis, or programmed cell death. The important thing about apoptosis is it does not elicit inflammation. Remember when our cells get damaged, right? They call out and we'll get sometimes an inflammatory response. In this case, we don't necessarily want an inflammatory response. Right? The cells just die. And they'll be cleaned up by resident macrophages nearby. Right? We don't need to call in the reinforcements. The other thing that'll happen, as we saw in that animation, is it will degrade messenger RNA. This prevents definitely the virus from being able to make its proteins from our machinery. It also causes changes in the membrane components of the cell. So hopefully, remember the first step of a viral infection is they have to do what to our cells? They got to attach. So if we change our membrane, those receptors might not be available to them to be able to attach. So we may be able to stop them from even getting in. And of course, if they can't get in, they can't replicate. We can stop this cycle. So remember, the cell that's infected, it's going to produce these. It may or may not be able to stop the virus. Right? But by producing interferon, it can give it to the neighboring cells. They're going to get ready. So they're going to maybe stop it from getting in by changing their membrane components. Or then once it gets in, they're going to degrade the messenger RNA it produces, or even stop protein synthesis, shut it down by stopping ribosomes from binding to messenger RNA. Ultimately, we're going to stop this cycle at some point. We're not going to be able to allow them to use us to replicate. In addition, that particular cytokine is actually a group of cytokines, right? Chemical messages that white blood cells use to communicate. It's going to cause a particular set of white blood cells to proliferate, to increase in numbers. And that are your natural killer cells. So as the name implies, right, they're really good at what? Killing, right? And they're really helpful in viral infection as well, and killing cells that are infected by viruses. So having them available, right? So we don't really have an inflammatory response. We go about this a little bit differently, right? When it's this internal invader called the virus. So that's it for today, especially since I'm out of time, huh?
Tell us when test 3 is.
Yeah, test 3 is not until the end of the semester. I already have it on the schedule, right? I should. It's November, like, 21st, something like that. It's not for a while. If that.